Hello, welcome to this virtual talk of our Network Control Symposium, where I'm going to show you our analysis where we use network control theory to investigate structure-function relationship in a clinical neuroimaging study. So the clinical population that we're looking at here is 22q11 deletion syndrome. It's a neurodevelopmental disease that's caused by a microdeletion on chromosome 22, and which comes with a very high prevalence of schizophrenia. And this makes it a model to study developmental alterations related to psychosis and also before the onset. So far, we have been looking at resting state functional network alterations, where we found disrupted triple network activity and a particular implication of the salience networks and some dynamic activity alterations. And what we wanted to look at next there is how does the brain structure support such aberrant dynamics? And to do that, we are using network control theory. So this approach is based on the assumption that the brain dynamically switches between different brain states. So here they're visualized with blue and red activity in the brain regions on top of a structural network that's here indicated by the black lines. And the second assumption is that this activity can be controlled through a control input at different nodes of the brain. In average controllability, which you will have seen in the talk before me, we look at control inputs on a single region and we investigate how well the brain could be transitioned to any other state from there. What we used here is a slightly different approach where we look at the transition between particular states. So through the calculation of minimal control energy, the network control theory framework allows us to see how, given control input at some brain nodes, at some brain regions, how much energy a brain state transition would require. We use a linear time invariant model and then optimize this cost function to estimate the optimal control input U, or if we, in case of this study, we used minimal control, so we just minimized the energy that would be required for the state transition based on this dynamic model. Here in this study, we extracted this structural brain graph from diffusion MRI and the brain states from resting state functional MRI. We assumed full control, so we assume that we can put an input control signal at every region of the brain. And in particular, we were looking at persistence control energy. So the initial state and the target state were the same. The functional brain states were retrieved using innovation-driven co-activation patterns. For more details on this, I will let you look at the references here. So basically, we decompose the resting state fMRI signal in different functional brain states. And then for each of these brain states, we computed the persistence control energy given an individual's structural connectivity. And in the first analysis, we wanted to see how this persistence control energy on real structural brain networks compares to randomized networks. And what we found is that in any of the 17 states that we look at, so this is on the x-axis here, the persistence control energy on the original structural connectivity matrices is always lower than on randomized brain networks. So the structural connectivity of the brain in any subject is wired in a way to minimize persistence control energy. And then also for the functional brain states, we randomized the maps, the spatial maps of these brain states. And also there, when we used random brain states, the persistence control energy was always higher. So the structural connectivity, together with the typical functional brain states that we find in the resting state MRI, has an architecture that minimizes this persistent control energy. That's something that has been shown before, and here we just confirm this finding. Then we wanted to see how this persistence control energy is altered in patients with 22q11 deletion syndrome. And we found that in particular in 3 brain states, 2 that are part of the default mode network and the silence network, the persistence control energy was reduced in these patients. And in another set of brain regions, including the amygdala and hippocampus and more posterior brain states like the pecuneus and the visual state, the energy was increased for these patients. That by itself was interesting, but then we wanted to see, now from the resting state fMRI, we know how long each of these states was active. So from the 8 minutes resting state, we can calculate from the time series of each of these states the activation duration. And we wanted to see how this activation during resting state relates to persistence control energy. So what we did, we simply correlated both of them across subjects for each state. And when we did that, unfortunately, there was no relationship. So that means that a subject whose energy is particularly high compared to other subjects does not spend more or less time in this particular brain state. On the other hand, when we computed the correlation within subjects, so across states, across these 17 brain states, we found that for almost all subjects, there was a negative relationship. So each of these individuals spent less time in brain states, in the resting state, that would require a low persistence energy based on their structural architecture. And interestingly, this negative relationship was weaker in patients with 20q11 deletion syndrome. This could indicate some kind of dynamic inefficiency of brain function. So to sum up, we found that the brain structural and functional architecture minimizes persistence control energy compared to randomized networks and states. I found that across subjects, persistence control energy and functional activation during rest are not linearly related, which is intriguing, but maybe it's just not, maybe it's just not specific enough for this kind of approach. Within subjects, however, we found that the brain activation minimizes control energy by spending less time in brain states with high persistence control energy. And this relationship is weaker in patients with 20q11 deletion syndrome. And with that, I would like to thank all the families who participated in this study, to my 2 former labs in Geneva, and to you for your attention.
Hi, my name is Linden Parks. I'm a postdoc at the University of Pennsylvania, working with Danny Bassett and Ted Satterthwaite. I'm going to talk to you today about a project that we've been working on comparing average controllability, which is a simple metric from network control theory, with strength from structural connectivity for predicting cognition. So each of the talks in this symposium are going to draw on the idea of network control theory. network control theory is quite a broad space, and each talk is going to use it quite differently. But for the most part, each of the speakers today, including myself, use this simple model described here. This model is a linear discrete time and time invariant model of dynamics, although in some talks you might see a continuous time variant, but it's broadly the same thing. This model is concerned with modeling the states of a system, the brain in this case, as they evolve over time. So let's break this down a little bit for you. X of t is an n by 1 vector that represents the state of the system at a given time point, where a state is simply a combination of brain activity in each of, say, n regions. A denotes an n by n adjacency matrix, which describes the structural connections between brain regions. In humans, we typically estimate this with diffusion imaging. B is an n by 1 vector that describes the brain region or regions that we want to use to control the dynamics of the system. And UK is a function that describes the amount of energy we inject into these control regions in order to influence the system. Now, if all of this seems a little confusing, I'm going to try and illustrate very plainly what happens in one of the simple metrics that we can get from this framework, which is called average controllability. So if you haven't heard of average controllability, I think it's best understood in terms of impulse response, which is to say we define a region of interest, then we can measure the extent to which that region distributes activity to the rest of the brain using its underlying connectivity profile. And average controllability sort of summarizes the spread of energy from a given region. So here I'm showing a toy network in the center of the screen, which represents our A matrix. It has 5 regions, which are all connected with weighted edges. Here, I'm only illustrating the weighted edges that immediately connect our region of interest, which is shown in this off-white color. But the rest of the edges in this toy network are also weighted. I'm just hiding them to keep the slide a little cleaner. So in this case, B over here simplifies to a vector where there's only one entry is equal to 1 and the rest of them are 0. And this entry of 1 represents our region of interest. And we're going to inject 1 unit of activity, which we store in U, just here. And we're going to watch the activity spread over time. But before we do that, we can also take note of some other properties of our region, which is that it has a degree of 4 and a strength of 1.08, which is just summing up these 4 weighted connections here. Okay, so now that we've set our initial condition, which we're calling T0, I'm presenting 2 toy networks here that only differ by some of the connections that are turned off in the right network. But notice that these connections are 1 step removed from our region of interest in the sense that we've preserved all of our immediate connections to our region of interest. This means that the degree and strength of our region are unchanged. Since the local connections are preserved, these lesions will impact the spread of activity though. So now we're going to step through units of time and we're going to watch the activity in each region change, which I'm also illustrating down here in these 2 matrices where the rows are the 5 regions and the columns are different time points and the cells are the energy, the activity in a region. So as we step through at the next time point, both networks still look the same, of course, because activity has only been able to sort of spread along those local connections. But as we keep ticking along more and more until we get to here, we can see that the impulse response or the activity spread is now quite different between our 2 networks. In particular, we can see that activity has diminished much more rapidly in the network on the right that is missing those distant connections. So the lack of those connections has basically restricted our region of interest's capacity to distribute energy throughout the system, and therefore it has a lower average controllability. So the take-home message here is that compared to strength, which is entirely determined by the local connectivity of a region, average controllability is influenced by local connections and the more extended connectivity represented in longer pathways from the region. This is relevant because if you have a region that has only a very limited local connectivity, which I'm kind of illustrating here on the left, then strength and average controllability will capture redundant information. But we know that this is not true of all regions, right? Instead, many brain regions have much more extended connectivity profiles, which I'm showing here, where regions connect to regions, which connect to more regions, et cetera, et cetera. In these cases, average controllability and strength will depart from one another because average controllability can tap into those that more extended connectivity profile of a region. So My interests are primarily in individual differences, so I was interested in examining the extent to which average control serves as a better predictor of cognition. So how I did that was I took roughly 1,000 participants from the Philadelphia neurodevelopmental cohort, which we have here at Penn. I generated adjacency matrices using 200 regions based off deterministic tractography, and I weighted the edges by streamline count for every subject. Then for every subject and every region, I calculated strength and average control. Then I used a kernel ridge regression to predict executive function. Now, just for brevity's sake, I'm not going to go into the measure of executive function today, except to say that it was a composite score from a computerized set of cognitive tests that we have here, and then we conducted at Penn with the Philadelphia neurodevelopmental cohort. So for each of strength and average control, I assessed prediction accuracy using nested 10-fold cross-validation and assessed accuracy using both the coefficient of determination, or R squared, here on the left, and root mean squared error here on the right. I repeated the nested cross-validation procedure 100 times, each time just using a different random split of the data. So each point in the distributions I'm showing here is one of those 100 splits, and each point represents the mean out of sample accuracy for each of the 100 splits. So we can see quite clearly that average controllability yields higher out of sample R square and lower out of sample root mean squared error, suggesting that it performs better at predicting our measure of executive function compared to strength. So next, we wanted to examine whether or not average controllability's capacity to capture the extended connectivity profiles of the regions was what was driving the better performance compared to strength. To check this, we turn to the principal functional cortical gradient, which we derived in our own data and is shown here at the top of the slide. We find a principal gradient that captures the canonical one, wherein transmodal cortex is represented here in yellow at the kind of top end of the cortex, and the unimodal sensory cortex is represented in blue at the bottom end, and then the other regions sort of transition gradually between those 2 points. The reason we really like this approach is because we find that it's a really great way of distinguishing between regions that are predominantly characterized by profiles of local connectivity and those that are characterized by more extended profiles. So on the left here, I'm showing the group averaged structural adjacency matrix from our own sample, but I've reordered the rows and the columns according to the gradient values. Then if I downsample this matrix into bins of 10 regions along the gradient, consecutive bins, and I calculate the strength of structural connectivity between every pair of bins, and that's what I'm showing in this middle matrix here. So along the diagonal, basically what I'm showing is the strength of connectivity between 10 regions that are within a bin, and then off the diagonal, I'm showing the strength of connections across pairs of bins. As you can see quite clearly, the regions at the unimodal end of the cortex here are characterized by strong connectivity between themselves and their nearest neighboring bins, but the connectivity drops off quite quickly as we move further up the gradient away from those regions. On the other hand, if we look at the regions at the top end of the cortex, the transmodal end, they have very strong connectivity within the bin, same as the unimodal end, but then they have sort of long direct connections down the gradient that also occur in a cascading manner. And I'm illustrating these here in these gray highlighted sections. So this illustrates that these regions of the transmodal cortex have these very extensive connectivity profiles that travel along long pathways. Okay. Furthermore, we find that the cortical gradient explains variability in cross-subject correlations between strength and average control. So each point in this plot on the right is one of the 200 brain regions from our parcellation. The y-axis is the correlation across our 1,000 subjects between strength and average control for that region. And you can see that when we, and the x-axis is the region's cortical gradient value. So you can see that when we look at regions that are at the unimodal end of the cortex, the cross-subject correlation is around about 0.8, suggesting that strength and average control are almost redundant measures of individual differences. However, as we move up the gradient towards the transmodal end, this correlation drops down to about 0.5, suggesting that while strength and average control are still highly correlated, they're no longer as redundant at the transmodal end of the cortex, and they have some unique variance, if you like. Okay, so to test whether this effect of the cortical gradient actually influenced our prediction accuracy in our main analysis, I took the 10 bins of regions along the cortical gradient that I've just mentioned, and then for each bin, I performed 10-fold cross-validation stratified on our measure of executive function. In this analysis, I also included a series of covariates, which I've listed here. Each of these were fit to the train data in each split and then applied to the test data in order to try and minimize leakage. So this basically allowed us to examine prediction accuracy as a function of the cortical gradient at this resolution of bins of 10 regions. Just also note at this point, because we used a nonlinear regression technique, the kernel ridge regression, we were unable to sort of simply just interrogate regional coefficients from a full model. This is why we took this approach. Okay, so on the left here, I'm showing this analysis for strength predicting cognition. And then on the right, I'm showing it for average controllability predicting our measure of cognition. In each plot, the black dots are the mean out of sample root mean squared error, and the vertical lines are the standard error over the 10 folds. We see that for both brain features, we get an improvement in prediction accuracy as a function of the gradient. Remember, this is error, so lower is better. But this effect appears more pronounced for average controllability. Down the bottom, I've summarized this effect using a Pearson's correlation, which is stronger for average controllability at negative 0.63. But just take these correlations with a grain of salt because they are estimated on only 20 data points because we only had 20 bins of regions here. So given this low power, I wanted to flesh out this effect just a little bit more. And to do so, I'm going to return to the toy network I showed at the start of this talk. Something I skipped over at the start of the talk was that prior to estimating regional average controllability, we normalize the adjacency matrix by every subject's adjacency matrix by dividing each element by the largest eigenvalue of the matrix and then scaling it by some constant c. And that's what this equation is here. By default, we set this to 0.99 and everything you've seen today use that default setting. Without going into details, setting C at 0.99 ensures that the activity spreading throughout the system decays over time. And you can kind of see this quite clearly in the toy network I showed at the start of the talk. You can see that the activity reduces and will eventually go to 0 over the x-axis if we ran it out for more time points. And just also note that if we were to set it at exactly 1, then the activity would never decay and just continue to circulate throughout the system. On the other hand, and more importantly, if we set C to less than 0.99, we basically force the activity to decay much more rapidly, which is what I'm showing here in these two simulations. For example, on the right, when C is equal to 0.5, we see that the activity only spreads around for a couple of time points before pretty much going to 0. This is useful because it means we can effectively tune the extent to which average controllability has access to the longer path lengths of a region's connectivity profile. Or put differently, we can effectively restrict average controllability to be mostly captured or mostly dominated by the local connectivity of a region, which should make average controllability look more like strength over our 1,000 subjects. And indeed, this is what we get. So here in the top left, I'm showing the results for the cross-subject correlation between strength and average control that I've shown previously, where the default was 0.99. And I'm repeating that analysis then in each of these plots, recomputing average control at different levels of C, which I'm showing at the top. So two things are obvious. First, on the y-axis of each of these plots, the cross-subject correlation pushes up towards 1 as we reduce C, indicating sort of an increased redundancy between the two measures. And you can see that up around here where most regions are well above 0.9 and pushing up towards 1. Second, the effect of the cortical gradient actually reduces. It gets weaker as a function of tuning C, starting at negative 0.5 and going to negative 0.38. Next, if we redo our prediction analysis using the cortical gradient bins, we see that the effect of the cortical gradient reduces from negative 0.63, which was our original analysis here, down to negative 0.19 as we reduce C. suggesting that the more we restrict average controllability to local connectivity, the less sensitive prediction is to the cortical gradient. Okay, to sum up, average controllability better predicts executive efficiency compared to strength. This better prediction appears to be facilitated by average control's capacity to capture the extended long distance connectivity of a region's connectivity profile, These extended connectivity profiles appear to be characterized by the transmodal cortex. And just some acknowledgements, first and foremost to my postdoc advisors, Danny and Ted, for creating a really wonderful environment to do my postdoc in, Professors Raquel and Ruben Gurr for their huge effort with the PNC data, which featured in this talk, my colleagues in this symposium, Urs, Daniela, Petra, and Fabio, some colleagues from the complex system group, Jason and Eli, and of course, funding from the NIH that supports my postdoc. And thank you guys for listening, and thanks to OHBM for, at the last minute, turning this into a virtual conference. And that's it.
Hello everyone, my name is Braun. I'm a researcher at the Central Institute of Mental Health in Mannheim and at the University of Pennsylvania. And in the following minutes, I want to demonstrate to you the utility of network control theory to investigate multimodal aspects of working memory. And in particular, I want to show you how you can combine methods from network control, genetics, pharmacological fMRI, and patient studies to disentangle dopaminergic contributions to the dynamic of large-scale brain circuits to working memory. To give you a little bit of background, working memory is a core cognitive function that encompasses the ability to maintain, but also to update and manipulate cognitive representations to successfully perform ongoing tasks. Proper execution requires the brain to transition through a diverse set of brain states, and on the left you can see one of the recent studies by Meg Shine and colleagues, in which they showed that the brain transitions through two different connectivity-based states during ever simpler performing memory tasks. There are a lot of other studies that show that indeed the brain needs to transition through various brain states that are globally expressed while performing working memory. But no one has so far looked at, or little has been known about how the brain actually controls these transitions and how it stabilizes these states. There's an interesting theory that actually tells us how that could work and binds it to dopamine, which is called the dual state theory of prefrontal function. And it posits that there are basically two dynamical states. One is based on D1 dominated state. That means preferentially D1 receptors are engaged. This state, or this dynamical regime, is characterized by high energy barriers between different states, and it needs a lot to actually go from one state to the other, so it's a really stable state. While the other dynamical regimes of the dual state theory says that if you have a D2 dominated state, then the energy landscape flattens and it's really easy to switch from one task to the others. We wanted to test these hypotheses or the prediction from the dual state theory in humans, and did so by using network control theory. The previous talks, you've already heard a little bit about the background of network control theory, so I'll keep it short here. We also start out with a multimodal brain atlas and do fiber tracking to get a structural brain network, and then we use functional fMRI to actually define the brain states. And we did so using a relatively well-known NBAC task. The NBAC task consists of basically two conditions in which you see a number of numbers appearing on the screen, and in the aerobic condition or the motor control condition, you just have to press a button which corresponds to the number you see on the screen, while in the 2-back condition, which is the working memory condition, you see you have to remember what you saw 2 screens before and press that button. So it basically has these two conditions called 0 back and 2 back, which correspond to motor or working memory content. We then used a simple GLM standard SPM approach and defined the beta weights corresponding to respective conditions as brain-wide activity patterns or brain states. Using network control theory, we then modeled how the brain transitions from one state to the other. Using optimal control, as you've heard before, this is something where you minimize the trajectory and the energy simultaneously. So that's why it's called optimum. And then we define 2 metrics. One is control energy, which measures how much energy or global energy the brain needs to go from one to the other. State and stability, which we defined as the inverse of the persistent energy, the energy needed to go or to stabilize one state. We then saw how we can use network control to track transitions to our working memory and looked at control metrics are associated with individual dopamine D2 and D2 receptor expression, and then we try to alter the system's dopaminergic state by using pharmacological fMRI, and finally we looked at alterations of controlmatic schizophrenia. We started out by looking at working memory in a set of healthy controls, and as you can see, we found that the stability of brain states, particularly the more cognitively demanding working memory state, is less stable than the 0-back state. This seems to be cognitively important because you can see that the stability of the 2-back state is associated with the performance of these subjects. So people that have a higher stability of the cognitively more demanding state also have a better performance during working, during end-back task. We then looked at how the brain transitions between those 2 different states and found that the control energy, so the effort it needs to steer from one state to the other, is also enhanced if you want to steer from the cognitively less demanding state to the higher demanding state. So the 0 to 2-back transition requires more control energy than the opposite transition. Looking at a regional level to actually identify the brain regions that are most important for these transitions, we found that there is a large overlap in these brain areas, which shows that there is a universal control set of preferentially prefrontal and parietal nodes that are important for both transitions, or that control both transitions. Having this framework that can model stability and transition between states, we looked at that if we can actually test the dual state hypothesis or the dual state theory, the predictions from it. As you remember, there are 2 dynamic regimes, one D2 related, which should be easy to switch between tasks, and a D1 dominated state, which should stabilize the system. And as we had no PET fMRI or direct measures of D2 and D1 receptor availability, we utilized a method that has been recently invented by the team from the University of Bari, in which they looked or developed a score that predicts the D2 receptor or D1 receptor expression based on the genetic profile of a individual subject. So you get a proxy of the D2 or the D1 expression in those subjects, and we acquired this score for our, computed this score for our subjects and looked if it correlates with the stability. And indeed, as we would predict, that D1 receptor expression, more expression of D1 is related to an increased stability of both brain states. For the opposite, we looked at how the D2 scores relate to the control energy, which should be that the control energy is negatively correlated or negatively predicted by the D2 outfit of patients or healthy controls. And as we can see, indeed, D2 score, the higher you have a dopamine D2 expression in the prefrontal cortex, the less control energy you need actually to switch between states. Having this framework established, we look if we could actually manipulate it towards less D2 dominated state using a pharmacological experiment in which we did a placebo control crossover trial with misopride. Misopride is a substance that is relatively selective for D2, or is a relatively selective D2 antagonist. And in line with our theory, we found that misopride increases the cost of controlling trajectories, so the control energy for both transitions increased. Dopamine dysfunction, working memory deficits, and alteration in brain network organization are hallmark of schizophrenia. So we also looked at schizophrenia to investigate how our network control metrics are altered. And as you can see, we found that the stability of brain states is altered in schizophrenia, but this is only in the cognitively more demanding tasks. So the 2-back state seems to be less stable, while the 0-back state is not different from healthy controls. Looking at the control energy, so the energy to actually control these switches, we found that the 0 to 2-back, so the transition into the highly cognitive demanding state is altered in schizophrenia, while the other transition doesn't seem to be affected. These results suggest that the brain energy landscape is more diverse in schizophrenia than in controls, and in line with the idea of a subtle randomization of brain networks, as Braun put it, which should make the system more difficult to steer appropriately. As you can see here, there's an illustration of that idea that the brain actually is that in the brain, the energy landscape for healthy controls, it is more pronounced in a way that it's easier to find an ideal trajectory, while in schizophrenia, this is the second part of the mind, this is more diverse or more flattened, and it's harder to find the optimal trajectory. To quantify that notion, we added subtle perturbation to the optimal trajectory by adding a random amount of noise to it, and then recalculating the trajectories to look how variable they are. And indeed, what we see, that the relative deviation of the trajectories is increased in schizophrenia, which is indeed in line with the idea that the energy landscape is more diverse, and that you can that the deviation from the normal trajectory can be induced by less or by really small perturbation to the optimal trajectory. To conclude, I hope I could convince you that network control theory is a useful tool to actually investigate cognitive dynamics in humans. I showed you that cognitively more demanding brain states exhibit a lower stability, and that accessing those states is more difficult, in line with the idea of associated switch costs when turning to more difficult tasks. I showed you that there's a universal set of brain regions, mainly located prefrontal and parietal cortices, that support these transitions. The ability to control brain dynamics in working memory is differentially modulated by dopamine receptor functioning in a way that D1 increases stability, while D2 signaling seems to lessen the effort needed to control trajectories. And schizophrenia patients showed a reduced ability to control and stabilize working memory patterns. Thank you very much.
Hi, so my name is Petra Vertes, and I recently started my own group in the Department of Psychiatry at the University of Cambridge. I thought I'd just start with an overview slide of my work. This is a word cloud based on my past papers, and you can see at a glance that the key themes are human brain networks. So at least that means I'm at the correct conference. But in parallel to this focus on human brain networks, I've also studied C. elegans cellular networks for the last few years. And I'm going to spend the next few slides or few minutes hopefully showing you why. In his Nobel lecture in 2002, Sidney Brenner called C. elegans nature's gift to science. And he started working on C. elegans about 30 years prior to that. And he chose that model organism for some very pragmatic reasons. So it's small, but not too small. It's just about visible to the naked eye. It's easy to grow. It's got a 2-week life cycle. It's transparent, so you can see what's going on inside. And importantly, it's simple, but it's not too simple. It's got 1,000 cells. And importantly for us, a third of those cells, so 300 cells, are nerve cells or neurons. So it's also quite a smart animal. Since then, of course, C. elegans has become the workhorse of many fields in biology. So in developmental biology, for example, it's an organism with a fully mapped developmental lineage for each cell, which makes it a very useful model system. It's also extensively used in genetics. And in fact, it's the first multicellular organism with a fully mapped genome. It's even used in space exploration. So it turns out that when looking through the wreckage of the Columbia disaster, it turned out that C. elegans cultures were the sole survivors of that mission. And they were used to study the effects of microgravity on living organisms. Closer to home, C. elegans is very useful in neuroscience. It's got 300 neurons, like I mentioned, and we know an awful lot about each one of them. So we know where they are located within the worm. We know their morphology. We increasingly know about gene expression inside individual cells. And for many of them, we also know their functional role. That's thanks to laser ablation experiments where you essentially remove or ablate neurons or classes of neurons and then observe the effects on behavior. Importantly, it's also the only organism for which we have an almost complete wiring diagram at the cellular level. So we know how these 300 neurons connect and talk to one another. And that makes it a gold standard connectome or network for network science too. And in particular, it's sort of inspired and corroborated many other analyses in human brain networks. So there's a large literature showing that both human and C. elegans brain networks share a number of organizational features in common. They both have modules, they both have hub nodes, small word property, hierarchy, and so on. In my own work, I've shown that C. elegans, just like human brain networks, also has a so-called rich club, which in C. elegans you can really demonstrate that this network property pinpoints functionally important roles. We also showed that both of these networks can be modeled using very similar generative models. And more recently, it turned out that principles of network control can be usefully applied both to human brain networks, and that's hopefully what the previous talks showed you, and to C. elegans, which is what this talk is going to be about. So it's important to note that despite its simplicity, C. elegans also displays a range of quite sophisticated behaviors. So it's able to sense attractive and aversive stimuli. It's able to move towards or away from them. It's able to reproduce, obviously, to sleep, to learn. It's even got a behavior called nictation, where the worm essentially stands up when food is scarce and looks to jump on the back of a Drosophila or another flying insect to fly to another place where food might be more abundant, which I think is amazing. An important question is how all of these behaviors are determined by the neural network. And so one fun way to address this is through robotics. So it turns out that you can build a little Lego C. elegans robot, and the instructions of this are open source, they're online. And together with my collaborator Danielle, we thought that it would be fun to try it out. So here's what you do. You build the robot and you attach two sensors. One is a sound sensor, which is going to mimic sensing food stimuli. And the second sensor will be a touch sensor or a sonar, which will mimic the C. elegans' ability to sense touch on the head or near the nose. And then the output, of course, in this system is going to be the wheels, or rather the motors that are driving the wheels. So here's how it works. You take the signal from these two sensors and you feed them into the appropriate sensory neurons in the C. elegans network. So we know which neurons are supposed to sense food stimuli, and so any input that is detected by the sound sensor will be fed into those specific sensory neurons. Then you let this signal percolate through the in silico representation of the C. elegans connectome, and you read out the signal arriving at the motor neurons, and you feed this output essentially to drive the motors, which then drive the left and the right wheels of the robot. So it turns out, if you do that, that this little robot is able to replicate two simple C. elegans behaviors. One is C. elegans moving towards a source of food that is detected, and the other is that the worm reverses when it senses a head touch. That's an aversive stimulus, so it reverses, sometimes changes direction and then carries on. So this is what you can see in this video of the robot. So you can see the shoes of my collaborator Gang. He's holding up his cell phone to the robot. I'm not sure whether you can hear the sound, but the cell phone is playing music, and that is causing the robot to move forwards as it detects the sound. In a minute, Gang will touch the sonar, and the robot will reverse and then keep going in reaction to this aversive stimulus. So I think that's quite amazing to watch, but of course there are a number of caveats. So this robot is only able to replicate two very simple behaviors, and more importantly, the network in the middle is really acting as a kind of black box. So it tells us nothing about the underlying neuronal circuit that is responsible for the behavior and that implements it in detail. Some C. elegans behaviors have been so extensively studied over the decades by experimental biologists that they have drawn up putative circuit diagrams. So the most famous one perhaps is the light touch circuit, and that's responsible for the worm reversing away from, in this case, an eyelash that gently touches an area near the worm's head. And it turns out that when you try to draw out this circuit, the sensory neurons that first detect the input are quite easy to identify. What's more difficult to identify is the role of the remaining 300 neurons in driving or facilitating the behavior. And so we don't really know whether the circuit diagram that we already have is complete or accurate. And also it would be useful to have some theoretical tools to guide the experimental mapping out of these circuits, because this takes decades to do, just experimentally. So we decided to try and use control theory to interrogate these circuits. And this is joint work with Bill Schaefer's lab. He runs an experimental lab on C. elegans neurobiology in Cambridge, and Laszlo Borabás's lab in Boston, looking at complex systems and controllability. Sorry. So just as a recap, we say that a system is controllable if it can be driven from any state initially to any desired final state in finite time. And the two pieces of information we really need are this matrix A, which is the connectivity or the wiring diagram of the network or the system we're trying to control. And then the equation here, which is the dynamics at each individual node. So how is the state of a node updated over time, depending on external stimulus coming in and the connectivity to the rest of the network. So we often define some driver nodes. These are the nodes that will be controlled by outside stimuli. And then the question is whether controlling just these driver nodes is enough to make the entire system controllable. And you can assess this using the Kalman rank condition, which is essentially by assessing the rank of this controllability matrix C. But that's very hard to check for large systems, more than 50 nodes. So you can also use the maximum matching framework, which makes it more tractable. But in any case, in the context of C. elegans behavior, this didn't seem to fit exactly. So for the driver nodes, we guess, or we know that these should be the sensory neurons, because they are the ones that are receiving external input and then driving behavior. But the goal isn't really necessarily for the entire nervous system to be controllable. Instead, we just want to elicit a particular behavior, which is a locomotor behavior. So what we're really trying to control are the muscles of the organism. And so for that reason, we made two modifications to the framework. Firstly, we added additional nodes that represent muscles. So some of our nodes in the network represent neurons in the nervous system, but we added also some nodes representing the muscle cells. And then we also use a target control framework. So instead of wanting to control the entire network, we only want to control the state of the nodes corresponding to muscles. And that modifies also the controllability criteria. So using this setup, we can then ask how the number of independently controllable muscles depends on the existence of each individual neuron in the whole network. So we look at neurons one by one or class by class, and we, in silico, we ablate them, we remove them from the network, and we ask whether the number of independently controllable muscles is affected by this deletion. And for the vast majority of neurons, we find that it makes no difference at all if you remove them. But we found some neuronal classes which did affect muscular control, and the number of controllable muscles was decreased. And it turns out if you tabulate these theoretical results, that we're reproducing a body of experimental work showing these same neuronal classes, which have been ablated experimentally with laser ablation, and which have led to uncoordinated motion or lack of locomotion altogether as a result of these ablations. There was one additional prediction, which was this neuron PDB, which hadn't been implicated in this behavior before. And so Bill Schaefer and his colleagues, Yilian Chu and Denise Walker, set out to verify these experimentally. And sure enough, they found that if you ablate this neuron, it will also lead to abnormal locomotion in this paradigm. So this was exciting for two reasons. Firstly, using control theory, we were able to make a new prediction in this really heavily studied, well-characterized biological system. And secondly, this seemed to be the first, I would say, maybe not validation, but proof of principle that principles of network control can be useful in making predictions in real neural networks. So as with any theoretical work or modeling work, we had to make a number of assumptions just to make the problem tractable. So we wrote a follow-up paper where we describe each of these assumptions in detail and kind of appraise the assumption itself and sometimes explore what happens if we deviate from the assumption. I don't have time to present all of that, but I thought I'd mention just the most controversial assumption, which is the assumption of linear dynamics at each of the nodes. And this is very unlikely to be strictly true, but it's an assumption that we need to make in order to make the problem tractable. And so our approach was, let's make this heavy-handed assumption, and then let's experimentally test whether this simplified model is still able to make useful predictions. So we found that it did. And I think this is somewhat surprising, but not entirely, perhaps for two different reasons. The first is that the vast majority of neurons in C. elegans are not spiking neurons. So that's a difference to compared to human neurons, for example. And so actually linear models of simple C. elegans circuits have previously been applied to successfully predict circuit functions. So the assumption isn't really that novel or maybe that shocking in the context of C. elegans. Secondly, kind of more theoretically, it's not really known how linear and nonlinear control are related for a specific network. But there are at least some cases in which the two seem to converge. And I think that's an interesting kind of theoretical direction to pursue in the future. As an echo of that, perhaps, there are also some simulations of neural networks with nonlinear dynamics, which were successfully predicted or replicated with linear control tools. So with that, I'd like to thank all my collaborators. So Gang Yang and Emma Towson in Laszlo Barabás's lab in Boston, Yilian Chu and Denise Walker in Bill Schaefer's lab in Cambridge, all of my colleagues in Cambridge, and you, of course, for your attention, as well as the funders. And I look forward to taking any questions in a couple of weeks' time. Thank you.